Hello, and welcome to section 8.3, Punnett squares. Tremendously exciting. Punnett squares are a very useful tool in genetics to help us predict outcomes of genetic crosses. When we mate two different organisms, what are we getting for our outcome? How do we know? Here's this question rolling around in Mendel's mind. How are we going to predict that? Well, let's look at how to make Punnett squares. So let's look at this example. I would like to take a look at the trait of stem height, tall versus short. And I'm going to take an organism that is tall and cross it with one that is short. And I know the genotypes there, homozygous dominant and homozygous recessive. What are my offspring going to look like, and how can I determine that? Here is how I set up my Punnett square. I'm only looking at one trait, so I'm going to create a grid that is 2 by 2. And that's how I'm going to set my Punnett square up. I'm going to first look at one of the parents. Let's take this parent first, the homozygous dominant parent. And I'm going to give that parent this side of my grid. I'm going to place one of each of its alleles over each column. Then I'm going to look at my other parent, my short parent, and I'm going to give that parent this side of my Punnett square. And I'm going to place one of each of that parent's alleles beside a row. Now I've just set up my Punnett square. Now I fill it in, and this is how I do that. I take this and this allele and I cross them. And the product of those, we always write the capital first, is that. I'm taking the big T crossed with the little t, and I get big T, little t. What do you know? Now let's do the same thing in this square. I took big T from this parent, little t from this parent. I get big T, little t. Now if I do that for all the squares, this is what I get. So I have just filled in my Punnett square with all of the different alleles and the different combinations. Now let's interpret that. Let's go to the next frame for that. How do I interpret this Punnett square? Well, here are my offspring. These are all my offspring. These are the genotypes of my offspring. Remember, the genotype tells the type of gene. How many offspring do I have that are the same genotype? They're all the same. I have 100% the same. They're heterozygotes, aren't they? They're heterozygous for height. So I can write that in. The genotype, T, they're heterozygotes, and I have 100%. All of my offspring will be that. What about the phenotype? What are they going to look like? Well, if you'll remember, when we look at this, the dominant trait is tall. So each one of these has a dominant allele. Therefore, 100% of all of our offspring are tall. That's their phenotype. So we can see that the Punnett square is a very useful tool to help us explain Mendel's results. And this is what our Punnett square showed. It just showed us that. When we cross these, we get all tall offspring. Now, what happens when we cross these two plants from this F1 generation? We cross two of these heterozygotes together, which is what they did right down here. Take two heterozygotes and cross them. Look what we get. We get all these different genotypes. Look, we get a homozygous dominant, we get two heterozygous, and we get one homozygous recessive for the different genotypes. How can two tall plants make 25% of the time a short plant? So out of all the offspring, we expect to see a short one 25% of the time. How does that happen? Well, we can use a Punnett square as our tool to help us explain why short plants reappeared in this second generation. right here. So let's do a cross between these two heterozygotes. Remember, we'll take one parent and look at its genotype. It's a heterozygote. And we'll give that parent one side of the Punnett square. We'll give it the top side, just arbitrarily. One of its alleles goes here and one here. Then let's look at the second parent. It's also a heterozygote. It will get this side of the Punnett square, and I will put one of its alleles here and one of its alleles here. Now, let's go ahead and cross these. When I do, I get big T, big T, big T, little t, big T, little t, and little t, little t. Now, let's interpret that. What am I getting here? I am getting, right here, one of the offspring is homozygous dominant. Write that down. One is TT. It's homozygous dominant, right? I have two offspring that are heterozygotes, and this is their genotype. And then I have one offspring that is the genotype little t, it's homozygous recessive. I'll just abbreviate that there, so I don't really have room to write it. So, in other words, my one time out of four, for every one offspring out of four offspring, I am getting the homozygous dominant. 1 out of 4 times I'm getting homozygous recessive, and 50% of the time I'm getting heterozygous. What are the phenotypes here? I just listed the genotypes. What about the phenotypes? Well, when I look at the phenotypes of these, this offspring will be tall. This will be tall because the tall is dominant over the recessive. This offspring will be tall. The tall is dominant over the recessive. This offspring will be short because it doesn't have any dominant tall alleles in it. So we have 75% of the time tall and 25% of the time short. And that's exactly what we see right there. 25% of the time we will get a short organism, a short offspring that is produced. Now I'd like to look a little bit at how we're reporting this. There are basically 3 different ways you can report this, and all of them are perfectly acceptable. You can simply count. So we can, here's the example we just worked out. We can count the different genotypes or phenotypes. So let's look at genotypes first, and we can say that for the dominant, homozygous dominant, to heterozygous to homozygous recessive, we can simply say it's a 1 to 2 to 1 ratio, and that's perfectly acceptable. The phenotypes in this case would be tall to short, and we could simply say those are in the ratio of 3 to 1, and that is a way that we can report the ratios. Another way we can report ratios is by using a fraction. So if we count genotypes, we can say that for homozygous dominant genotype, 1 out of 4 is homozygous dominant. And heterozygous, we have 2 out of a possibility of 4 total. So 2 out of 4. And then for the homozygous recessive, we only have 1 out of 4. And then the same with our phenotypes. We could say tall would be 3 out of 4. And we could say short would be 1 out of 4. And that would be one way of writing the, interpreting the results. and writing the ratios as in fraction form. Another way we can also do this would be percentages. So you know that if you look at, just looking at this square, there are four. Everything's 1/4 is the same as 25%. If you type that into your calculator, 1 divided by 4 gives you 0.25. Multiply that by 100 to get your percent. And you get homozygous dominant, 25%. Heterozygous is 50%. And homozygous recessive is 25%. And you get your tall at 75% for the phenotype, and the short would be 25% for the phenotype. So any of those ways are perfectly acceptable. If you like counting, if you like fractions, if you like percentages, those are all acceptable ways to write the ratios for the interpretation of the Punnett squares. Now here's a little example for you. Set up a Punnett square to represent this cross, homozygous dominant, big T, crossed with this parent, a heterozygote. What will the genotypes and phenotypes be that result? So I'd like you to pause the video, see if you can do it on your own, try to set it up, and go ahead and start the video again, and I'll solve it for you. Okay, let's go ahead and set this up. We have one parent right here, the homozygous dominant, and I'm going to give that parent this side of the Punnett square. And let's give this parent the other side. So I'm putting this parent's alleles right there. And I'm going to go ahead and fill these in. Those are all of the offspring that we get. And let's interpret these results. I'll just write these in fraction form. Homozygous dominant genotype. Let's look at the genotypes first. I have one here and one here. That would be 2 out of 4. Now let's look at the heterozygotes. That would be here and here. That is, again, 2 out of 4. We could also write those as 50% and 50%. Let's look at the phenotypes. If we look at this, this will be tall, 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 and tall. I have 100% tall for my phenotypes. And here's another practice problem for you. So after I read it, go ahead and pause the video, see if you can set this up on your own. And then I'll walk you through the answer. So draw a Punnett square now to represent the cross between a heterozygous red-flowered plant and a white-flowered plant. Those are the two separate parents here. Heterozygous red-flowered and then a white-flowered plant. And we are to assume that red is dominant. So I'm going to arbitrarily choose a capital R for red. That will be my dominant allele. And then I'm going to say that lowercase r will be my recessive allele, and it will represent the white phenotype. If I do that, I first now need to look at my heterozygous red flower plant. That's going to be my first parent. So my first parent is a heterozygote on this side of the Punnett square. Okay. My second parent will be the white flowered plant. And in order to be white, it has to have that genotype, little r, because if there is a big R, then it's going to be red. So it has to be homozygous recessive in order to be a white flowered plant. Now I'm going to go ahead and fill in my Punnett squares. Remember, I'm just crossing these. I have the R down from here and the little r from there, and I can cross those. Now what are my genotype ratios? I have this for a genotype, 50% of the time. And then I have homozygous recessive, 50% of the time. What are my phenotype ratios? Well, red will be 50%, and white will be 50% as well. I have red here because I have a dominant allele, and red here. And then here and here, are white because there are no dominant alleles present in those genotypes. A few more items to note before we end this section. Letter choice. When you choose letters, just keep in mind that letters such as W are really hard to distinguish a capital from a lowercase. A letter like D would be much easier, or R, where you can actually distinguish them from each other. S is another really bad one because it's hard to tell when you start filling in your Punnett squares whether it's a capital S or a lowercase s. So try to pick letters that look a very different shape from each other, from the capital to the lowercase. Also, we always put the capital letter first when we are writing our genotypes. And just remember, homozygous recessive means that it's the two recessive alleles in the genotype. Homozygous dominant just means the two dominant alleles are the genotype. A heterozygote means one of each. It means one dominant and one recessive. That's a heterozygote. And then just remember the difference between genotype and phenotype. Genotype are the type of genes. Phenotype is the appearance. Here are some advanced ideas for you to consider if you would like to look at that. And I look forward to seeing you in class.